are ready to start a new session of next-generation enzyme replacement therapy. And we will have 4 presenters, and they will show you the results of clinical trial. As I mentioned before, I hope, because it is a company-sponsored trials, I hope you will take it with a grain of salt. Tiffany asked me to give a very brief 5 to 10 minutes introduction to this session and to very briefly explain how enzyme replacement therapy works. And I realized that to do this, I have to cover major discoveries in the lysosomal field in the last 70 years. So this story, the story began in the 50s when Christian Deduve described a new particle in the cell which he called lysosome. He later, by the way, hated this name because it was very similar to lysozyme, but anyway, the name stuck, and so this is a new particle, lysosome. What was interesting and quite remarkable that he made a link between this new particle and a process which he himself described and called endocytosis, a process by which cell internalizes the substances from outside environment. Furthermore, he made the prediction that this would eventually lead to enzyme replacement therapy. Here is what he said. Oh, I can't read it. I need someone help here. Okay. Oh, can I have some?
We can see it, Nina.
You can read it.
We can read it.
Okay.
More or less.
Yes.
If someone can change the font,
I guess I need my postdoc, so he'll push the buttons and everything will work.
Tell them to come up here.
Anyway, the text is kind of an important one here.
Where's the computer guy?
Can you make it a bigger screen here? So that I can see it, then I can read it, so that this will be a large screen, the whole screen.
I don't think they can do that, Nina.
Anyway, okay, so be it. The prediction was that the substances which come to inside the cells from outside will eventually end up in the lysosome, and this has a very serious consequences for possible interactions between the molecules and potentially for therapeutic intervention. However, at that time, it wasn't clear what is inside the lysosome and what is the role of lysosome in healthy cell. Until the next step, when the Belgian scientist Henri Urs discovered the lysosomal enzyme, which he called acid maltase or acid alpha-glucosidase, and he realized that this enzyme is missing in patients with glycogenosis type 2, Pompe disease. As a clinical entity, the disease was described some 30 years prior to that. He also made a prediction that there will be other lysosomal enzyme and their absence may cause other diseases. And in fact, Pompe disease was the first lysosomal storage disorder described, and now we have around 70 lysosomal storage disorders. The next major discovery came as an accident, and it happened in the lab of Elizabeth Neufeld at the NIH, when a postdoc in her lab, by accident, mixed 2 cultured cell fibroblasts, coming from two patients with two lysosomal storage disorders. And what happened was that these two cells corrected each other. It was quite a discovery made by, as I said, by accident, but it indicated that the enzyme can be secreted, and importantly, that they can be taken up by the neighboring cells. Since then, there was a huge number of papers and studies trying to understand how actually these enzymes work, what's the basic biochemistry of this enzyme, and how they are trafficking from where they are made within the cells to the lysosomes. And mostly in the lab of Stuart Kornfeld, it was shown that the enzyme is synthesized in the cellular compartment called ER. Then they glycosylate it, they move to a Golgi, and here they acquire mannose-6-phosphate groups, which serves as an address label to go to the lysosome. This mannose-6-phosphate modified enzyme is picked up by the receptor. The receptor brings it to the pre-lysosomal compartment, and here the receptor and the enzyme separates. They say goodbye to each other here, and the receptor recycles back for the next round of cycling, and the enzyme is discharged into lysosome where it is active. It was also shown that this is the majority of the enzyme made in the cell, but a certain portion of the enzyme, which is synthesized, is secreted, goes outside the cells, and it can be picked up by neighboring cells or by the same cells, because this mannose-6-phosphate receptor are also present on the plasma membrane. So this was the discovery of the mannose-6-phosphate mediated trafficking, mannose-6-phosphate pathway, and this was essentially a jackpot, because that's where people realized that if you have a recombinant enzyme properly made, it can be picked up, but mannose-6-phosphate receptor gets to the same pre-lysosomal compartment, endosome, and then can go to the lysosome and correct the defect in the enzyme. And in the 70s, again, this discovery was made at the NIH by Roscoe Brady, who identified the enzyme in Gaucher disease, the most common lysosomal storage disorder, and he was the first to initiate the first enzyme replacement therapy in non-neuropathic form of Gaucher disease, and it was a spectacular success because the patients now can essentially live normal life. And it is this success of the enzyme replacement therapy for Gaucher disease which stimulated efforts for other lysosomal storage disorders. And enzyme replacement therapy with recombinant human acid alpha-glucosidase made its way into the clinic in '99 when the first clinical trials began.